before getting started, I just want to remind you that you read a short section on UV in the disinfection chapter. So go back and reread that again now. It'll make more sense. So let's start off by looking at what UV light is. So UV light is electromagnetic radiation, and it's the spectrum that's just below visible light. So here we see visible light going from 400 to 700 nanometers. And UV is divided, it's just shorter than that, and it's divided into A, B, and C. UVA is 320 to 400 nanometers, UVB is 280 to 320, and UVC is 200 to 280. UVC is the most germicidal, meaning it's the most damaging to cells. So fortunately, and it is present in sunlight, so we wouldn't be here if it wasn't for the ozone layer, which absorbs almost all of the UVC irradiation and also most of the UVB irradiation. So sunlight really contains a decent amount of UVA and just a tiny amount of UVB. Those wavelengths are actually still capable of causing some damage to cells, and we'll talk about that when we talk about sunlight disinfection in a few lectures. So the way that UV disinfection works in treatment plants is that we generate these UV wavelengths that are very potent at causing disinfection, and we generate them with lamps. And so the most common lamp design is the one shown at the top. This is a low pressure mercury lamp. So it's a long tube filled with mercury and an inert gas, and there are electrodes at each end that produce an electrical discharge, and the mercury electrons in the mercury atoms get excited, and when they relax, they release photons. If we look at the next slide on the top, you can see here a spectrum of a UV mercury low pressure lamp, and you can see that it emits a very narrow band of photons with a peak at 254 nanometers. The other common lamp technology is medium pressure lamp, although I shouldn't say it's that common. So low pressure lamps are very, very common. Medium pressure lamps are in use, but they're much more expensive. They're more expensive to manufacture because they have to hold the mercury at a higher pressure. So they exist, but they're not used that commonly for drinking water or for water disinfection. And what happens in the medium pressure lamps is that when the mercury is excited at higher pressure, there are more energy states for the electrons to get excited to, and so when they relax, they release photons across a much wider range of wavelengths, both shorter and lower energy and higher energy photons. And so you would expect that that might have a different biological effect on cells, and indeed it does. Just going back to the lamp slide one more time, I just wanted to point out that the UV lamp technology is actually very similar to fluorescent lamps. Fluorescent lamps are also based on low pressure mercury, and the difference is that the sleeve is different. So the sleeve in UV lamps has to be quartz because regular glass absorbs UV 254. With fluorescent lamps, they use glass, and then there's an additional coating, that they always look white. That's a phosphor coating, which converts the UV 254 into visible light wavelengths. And because they can use glass, fluorescent lamps are cheaper than UV lamps. Okay, I'm going to just check to make sure that this recording is working, and it is not. Well, it turns out it was recording, so let's keep going. Let's talk now about the mechanism of damage. So what we have here is the absorption spectrum of an average, or I should say an average absorption spectrum of DNA and of a complex protein that contains many all the different types of amino acids. So only some actually are chromophores. So it contains all of those. And what is really interesting to see, and the reason why low pressure UV disinfection works, is because here at 254 nanometers, where the red line is, that coincides almost perfectly with the absorption maximum of DNA. So when the DNA absorbs light at that wavelength, so the DNA absorbs light efficiently at that wavelength. You can see that proteins don't absorb light that efficiently, but they still do absorb photons of that wavelength, and so it is still possible to get damage. You can see that proteins absorb more efficiently at longer wavelengths over here and at shorter wavelengths. So you might expect that medium pressure UV, which emits those other wavelengths of light, to be more effective, or to have, not to be more effective, to have different biological effects than low pressure UV254, and indeed it does. So here, just to give you a sense for what this technology actually looks like in practice, here's a UV bulb array. You can see a lot of UV lamps are used, and so this entire array will be inserted into a channel of water, which then flows parallel to the lamps. Now let's get into talking about the mechanism, and then we'll get into the results from the paper that you read. So here we have double-stranded DNA, and when a photon comes in, it can be absorbed by the pyrimidine bases. So here we have two pyrimidine bases that are adjacent to each other after they've absorbed UV, and they are now covalently bonded. And when that happens, it disrupts the double helix. Let's zoom in a little bit more to remind you what that double helix looks like. So here we have, you can see two pyrimidine bases. And as a reminder, up at the top, you can see which the pyrimidines are cytosine and thymine. So in their normal configuration, there are hydrogen bonds with their complementary base on the other strand. Okay, here's even more reminder of what the pyrimidines are. So in RNA, it's uracil, and then thymine and cytosine in DNA. And then this is similar to an image in your textbook showing a thymine dimer, but what I like about this figure is it also shows the phosphate backbone. Okay, so here's the DNA backbone, and then here's the dimer. So you can see that these are two thymines that are next to each other along the backbone, and then they covalently bond, either in this cis-syn configuration or over here in a 6,4 configuration, which I think is called a cyclobutane dimer, but that's not so important. The important point is that I just really wanted to emphasize that they're next to each other. Okay, so this covalent bond is next to adjacent thymines or any two adjacent pyrimidines. And when that happens, it disrupts the hydrogen bonding. And then this DNA can no longer be replicated. And presumably that's what leads to an activation. And we'll get to that. All right, so now before diving into the paper, I just wanted to remind you how UV dose is quantified. So we did this way back at the beginning of the semester in one of the cholera lectures, but let's go over it again. So dose is a function of irradiance times time, and irradiance is typically measured in milliwatts per centimeter squared and time in seconds. So when you multiply them together, remember that you can write a watt as a joule per second, and then the seconds cancel and you get millijoules per centimeter squared. Now, somewhat bizarrely, the textbook does not make that simplification. And furthermore, they represent the irradiance in microwatts per centimeter squared. So you end up with microwatt seconds per centimeter squared. Those are not common units. The authors of the textbook are microbiologists, not engineers. It's very uncommon to see those units in engineering literature. I also wanted to point out that the term dose is actually not the preferred, it's not the most technically correct term. The most technically correct term that's consistent with other types of research on the effects of light is fluence. But engineers prefer the term dose because it's sort of consistent with what's used for chemical disinfectants as well. So we'll continue to use that term. But note that fluence is also used to represent the same quantity. And then finally, down here at the bottom, millijoules per centimeter squared are the most common units used in the US, but in the rest of the world, it's more common to see UV doses represented as joules per meter squared. And I wanted to tell you that I did this conversion for you in that earlier lecture, and I actually did it incorrectly, which is really embarrassing. I converted centimeters squared to meters squared incorrectly. So if you go back and look at that, hopefully you'll see my error. Maybe you noticed it before and just didn't bother to tell me, but what's in this slide is correct. So you should go back and walk through that conversion to make sure I did it right this time. Okay, so now let's dive into the paper that you read. So this is a really nice paper. It's getting kind of old, but I still like to use it because it provides such nice insight into the mechanism of UV damage. The researcher who did this work is Kumiko Oguma, and this was part of her PhD, and she's now a professor in Japan and continues to do research on UV technologies and disinfection mechanisms. So if you like this paper, you might want to look up some of her more recent research. Okay, so in order to understand her work, it's really important to understand this actual assay that she developed, the ESS assay, the endonuclease sensitive site assay. Endonucleases are enzymes, and there's a whole class of them, and they cut DNA at specific locations. And so she used an endonuclease that cuts DNA wherever there is a pyrimidine dimer. And so in her experiments, what she was able to do is when E. coli or Cryptosporidium were exposed to UV light, if dimers formed in the DNA, then when she isolated that DNA and then exposed it to this specific endonuclease, the endonuclease enzyme cut the DNA wherever there was a dimer. And as a result, the strands of DNA got shorter and shorter and shorter. So the more dimers, the more cuts, and the shorter and shorter strands of DNA. So that's basically it. If you get that, then that's as much as you really need to know to understand how the methods worked and then appreciate the results. So let's look at panel A now. This is a photograph of an electrophoretic gel result. And so in the first two lanes, we just have the marker DNA. So that just tells you how far DNA strands of known length will migrate under the electric field. And then in the third lane, we have the E. coli DNA that's been extracted that hasn't been exposed to DNA. So you can see that it all clumps together at the very beginning. It doesn't migrate very far because it's very long. And then as the UV dose increases, so when we have 2 millijoules per centimeter squared, you see this smear because a bunch of shorter strands, different lengths, have been formed and they migrate further on the gel. And then as the dose increases, the dose was increased, got even more smearing and strands migrating even farther. So if you look on the right, this is just summarized nicely and also shows the DNA length and base pairs at the bottom. So this is strong evidence that as the dose increases, you get more dimers formed. I also just wanted to make a point about the doses. So she found that a dose of 2 millijoules per centimeter squared resulted in 90% inactivation when she measured inactivation just using a common culture-based method. What I reported earlier in the semester was, I think, a dose of 3 or 3.4 millijoules per centimeter squared. So those are a little bit different, but not very different. So that's kind of typical that every research study might produce slightly different results, but they're similar to each other, not too far off. Okay, so then she went on to relate the number of dimers, or these ESS sites that got cut, per base to the dose. So this is the number of, the y-axis could be interpreted as the number of dimers per 10,000 bases. And you can see that the increase is very linear with the UV dose, which, if that's the main thing that's going on, is what you might expect. So that's pretty neat. And what's really neat is that the number of dimers per base was related in a log linear way to the log inactivation. So on the y-axis now, we have the colony forming ability ratio, which you could interpret as the log reduction. So 0.1 is 1 log reduction, 0.01 is 2 log reduction. And you would expect a log linear relationship. Okay, this, what's shown on the y-axis, that's been converted to, or that's in a log base 10. This is what you would expect for a first order process. Okay, so this is very strong evidence that formation of pyrimidine dimers by UV is what actually causes inactivation of the E. coli. And then the second part of what they did in this research was then measure if those pyrimidine dimers could be repaired. And there are two classes of repair. There's dark repair, which is anything that doesn't require light, and then light repair, which not surprisingly requires light. So in this figure, you can see an example of dark repair. This is called excision repair. So the first thing that happens is an enzyme removes the section of DNA that's damaged that contains this thymine dimer. And then a polymerase enzyme then comes along and replicates the other strand, so puts new matching base pairs in. And then another enzyme comes in and makes the final bond, and then that double strand is repaired. And what's neat about light repair, which you can see on this slide, is that it can all be done by one enzyme. So this photolyase enzyme just comes and sits down on that thymine dimer, and then it has chromophores and absorbs visible light, and it uses the energy from those photons to break the covalent bond between the adjacent thymines, and then the DNA is repaired. Okay, so both of these types of repair are theoretically possible and well documented in general, and they wanted to understand how significant they were in E. coli. And then here's a molecular model of the photolyase enzyme, just because it's kind of neat. It's been very well studied. So what researchers found is that, look at the panel on the left now. Here's the result for 6 millijoules per centimeter squared, lots of short strands of DNA. And then as that sample was exposed to longer and longer periods of fluorescent light, many of the shorter strands get repaired, and you end up with lots of longer and longer strands. And so that is clear evidence that this photolyase enzyme is repairing many of these dimers. What's really neat again is that they were able to correlate, or you see a clear relationship between the decrease in the number of dimers and an increase in the number of viable E. coli. So if you look at it, and again, in terms of log inactivation, they were able to, or after 120 minutes of exposure to fluorescent light, let's see, 10% of the E. coli were viable compared to initially only 0.1% of the E. coli were viable. So all these E. coli in between regained their culturability. And then in the dark, they did not see this phenomenon. So they did not see repair of the dimers, and they did not see E. coli regaining their ability to grow in the lab. Okay, so that's the E. coli results. And I am going to pause one more time and make sure that the recording is working. Okay, looks like we're still recording, so I'm just going to keep going. Okay, so they did a similar experiments with cryptosporidium, and they also found evidence that dimers were formed and that those dimers were correlated with the UV dose. But, and then they also gave the E. coli time, the cryptosporidium time to repair, either in the light or in the dark, and they observed repair, but they did not observe evidence that the oocyst became infective again. If you want my personal read of the results, I think that they're not very straightforward to interpret because they had to rely on this mouse infectivity assay because the cryptosporidium cell culture assay wasn't that well developed at the time, and it's still not very many labs that can do it. But they will go with their results. I think others have sort of found comparable results subsequently. Okay, so that's pretty interesting, and what that would indicate is that there's additional types of damage that occurs in cryptosporidium. So even though the dimers are being repaired, there's other types of damage that are not being repaired, and so the oocysts stay inactivated. All right, so if you had questions on the Oguma paper, please hold on to them, write them down, and then pull them out when we meet again next week, and we can review any remaining questions that you still have. And now we're going to move on to looking at the susceptibility of different types of pathogens to UV. So what I have here is a graph that's a summary of a whole bunch of UV data from this paper that was published about 10 years ago with different organisms showing 1, 2, 3, and 4 log inactivation. So if we look up at the top, we see the organisms that are most susceptible. And here we have different bacteria, and then Giardia and Crypto. So as we've been saying all semester, Giardia and Crypto are quite susceptible to UV, so it's a great strategy for inactivating them. And in the middle, we see our first virus, hepatitis A, so still pretty susceptible. We've got polio, Coxsackie, and then when we get down here, we see adenovirus, which is actually quite resistant to UV. And then acanthamoeba, which is a free-living amoeba that grows in distribution systems. It's not necessarily that big of a health concern itself, but it can host Legionella, so it would be nice if we could inactivate it. Here we've got more adenovirus, so different adeno strains. Here we've got a bacterial spore, here another adenovirus study. So these are just different studies where they used different sources of adenovirus and different strains. And then Bacillus subtilis, another spore. So what we see is that there are that bacterial spores are pretty resistant, and that's attributed to absorption of the UV light by their cell walls, by the spore wall, and adenovirus. And if you compare like this result here of adenovirus type 40 to Giardia crypto or some of the bacteria up at the top, I mean it's highly resistant. So this is a big bummer because adeno is present in most wastewater, and UV has become a common strategy for disinfecting both wastewater and drinking water. But since adenovirus is so resistant, it's not it can't be used as the sole disinfectant unless you use really high doses. Since we're interested in mechanisms, it's natural to ask, well, why? What is it about adenovirus that makes it so resistant? And what's understood now is that adenovirus is a double-stranded DNA virus. You're going to get a presentation on this, or maybe you just did today or on Thursday. I can't remember which day it's assigned for. So adenovirus is a double-stranded DNA virus, and it can hijack, when it gets inside its host cell, it can hijack the repair mechanisms of its host cell. So remember, viruses wouldn't be able to do repair on their own because they don't have any machinery of their own. But because adenovirus has double-stranded DNA, which is the same type of DNA that its host cell has, the repair mechanisms can also fix the damaged adenovirus DNA. What I have shown here are results from a neat study done by Carl Linden's group at CU Boulder, where they show the log inactivation of adenovirus on the top by low pressure UV light, and then in the lower line by medium pressure light, UV light. So you can see that the medium pressure causes more inactivation. And the reason they think that occurs is because they think that medium pressure light, remember medium pressure UV lamps produce UV light at over a whole range of wavelengths. And they think that it causes other types of damage that cannot be repaired, in particular protein damage. So in the bottom graph, they're showing the number of DNA lesions per dose, and that's actually not that different for low pressure and medium pressure, and so they attribute the difference to damage to protein, and that can't be fixed. So this result with adenovirus is probably similar with other double-stranded DNA viruses as well, that they're quite resistant to UV. Ascaris eggs, it turns out, are also really resistant to UV. So this is work that one of my first students did, where we exposed Ascaris eggs to UV light. And I'll just draw you right toward the important result here. So if we look at this point right here, this is 1 log inactivation at a dose of 1,000 joules per meter squared. So we have to do our conversion here. So that's equivalent to 100 millijoules per centimeter squared. So I go back to that same graph, and now I've added Ascaris eggs down here at the bottom. So we can see that they're even more resistant than adenovirus. So not so effective. Now let's move on to talking about how this has affected actual practice. So remember that the way that the surface water treatment rule works for drinking water is that you have to achieve a certain numbers of log inactivation for Cryptosporidium, Giardia, and viruses. And to help you determine what if you want to use UV as your disinfectant, there are these tables, these lookup tables, where you can say like, okay, according to the surface water treatment rule, I need to achieve 1 log inactivation of cryptosporidium. Oh, that's great. I only need a UV dose of 2.5 millijoules per centimeter squared. But if I also need to get 1 log inactivation of viruses, then I need to apply a UV dose of 58 millijoules per centimeter squared. So this box here in blue, these are the UV doses that you need to apply to get your virus credits, and you can see how much higher they are than for viruses than they are for crypto and giardia. And so the virus dose is basically determined by adenovirus, and that's why they're so high. So as a result of this, UV disinfection of drinking water is typically applied in combination with chlorine. So you use UV and a lower UV dose than is shown in the blue box, and to use UV to get your credit for crypto and giardia, and then you use free chlorine to get your credit for virus, because viruses are still believed, or viruses are known to be quite susceptible to free chlorine. The requirements for wastewater are not as stringent, and currently most wastewaters do not have to meet a virus disinfection standard. And so UV disinfection is actually quite common for wastewater. One of the reasons is that if you use chlorine, and then you discharge your wastewater to a sensitive receiving body, you often have to dechlorinate. Whereas if you apply UV, you don't have to worry about that. And the UV doesn't leave a disinfectant residual. And so that can actually be an advantage. So UV has actually become quite common for disinfecting wastewaters. It's more common for disinfecting wastewater than it is for drinking water. Okay, so there are a lot of interesting research areas, ongoing research areas on UV disinfection, and so I thought I'd mention a few of them and might inspire some of you to pick something like this for your literature review. So the mechanisms of damage by medium pressure UV, or there's another technology, pulsed UV, which has its own unique spectrum of light that it produces. Those are hot areas for research right now, so that would be an interesting thing to explore further. There's also another even newer UV technology becoming available, and that's light-emitting diodes. So light-emitting diodes in the UV range have recently become available commercially, although they're more at the high end of the UVC range or the low UVB range. Like 260 nanometer LEDs are still quite expensive from what I understand, but the cost will probably come down a lot in the future, and so that's kind of an exciting emerging area of technology. And then finally, there's some work going on showing that different organisms appear to respond differently to different wavelengths. So you know, that summary table that shows the sensitivity of organisms at 254, you know, the order of those organisms may change at other wavelengths or with combinations of different wavelengths. So that's kind of interesting. And if you think about UV LEDs, you could potentially pick different wavelengths if you knew that certain wavelengths were more effective than others, or if there's some possibility that there could be synergistic effects between different wavelengths. And you could potentially design a UV disinfection reactor with exactly the combination of wavelengths that you want to get the most disinfection. All right, so we'll finish up by just showing you a few slides of what UV disinfection looks like in practice. This is a picture of a UV disinfection reactor at a wastewater treatment plant out in Contra Costa County, and all you get to see is the control panels with the electric cords going down to the arrays. So the arrays are underground here, like the array I showed you at the beginning, and the water's flowing underneath, and it's all covered because UV light is very bad for us, and so you don't want to get exposed to it. It seems like it's kind of a sophisticated technology, but it's actually not. It's actually quite easy to use, even in small installations. And so here's an example of a waste stabilization pond in New Zealand, and they're disinfecting the effluent with UV, and it's just this one single channel here. And so what's nice about this, again, compared to chlorine, is that chlorine you constantly have to replace your chlorine chemical, and then you have to be dosing it correctly, and there's a disinfectant demand, so you might have to be adjusting the dose. Whereas with UV, you just turn it on, and well, it's not quite that simple. You actually, ideally, should be measuring the transmittance of your water and making sure that the dose that you're delivering is the dose that you think. So if the absorbance of your water goes up a lot, meaning your water starts absorbing a lot of the UV light, then the delivered dose is going to go down. So there are simple sensors that you can use that measure the transmittance of your water to ensure that you are delivering the desired dose. And then finally, I wanted to present an example of a project that I've worked on for many years, where we developed a point of use device for household treatment of drinking water that's intended to be low cost and to work in low-income communities. And the design for this device is it's a tube of stainless steel with a UV lamp suspended over it, so you can see the lamp here, and the water just runs underneath it. And the reason we designed it this way is that there are commercial, there are small devices available for disinfecting water, but they're all pressurized tubes, and the lamp is submersed in the water. And when you do that, it makes it much more expensive for a number of reasons. So when you submerge the lamp in the water, you have to put a quartz sleeve around it to protect it, and that quartz sleeve gets fouled by constituents in the water. You can even get bacteria and algae growing on it, and you can also get chemicals precipitating on it. And so this is a big issue in UV installations. They actually have like squeegees that come and clean the lamps, and then you periodically have to take your lamp array out and submerge it in a bath of chemicals to remove the scalants. So by suspending the lamp over the water, you avoid having to do that. And then you also don't need to isolate the electronics from the water. Okay, so here's what the basic design looked like, and here's what the actual final product looks like. We call it the mesita azul, or the little blue table in Spanish. And so here is the UV tube, the stainless steel tube. And the way this device works, and here are the components over here, as you can see, the stainless steel tube, the UV lamp, and then the inlet structure that's designed to disperse the flow evenly and get nice laminar flow under the lamp. And here's the outlet structure, the outlet weir that controls the water level. And this device can treat 5 liters of water per minute. It uses a 15 watt UV lamp, which costs, last time I checked, about $12 to $15 usually, and we estimate could last for many, many years for a single household. And then a place to store your water. So the way this works is this was designed for households in Baja California, though it's now being used in other parts of Mexico, that go and collect their water manually. So they bring back this 20 liter or 5 gallon bucket of water, and they bring back their water in one bucket, pour it into this bucket, and it has a cloth filter here just to remove anything that's large. And the water in this region is pretty clear, not a lot of turbidity. So it's well suited for treatment by UV. and they turn the lamp on in their mesita azul, and then they open the spigot here, and the water begins to flow through the UV tube, gets disinfected, and then goes into this storage container. And it's really important that the storage container be what we call a safe storage container. In particular, because UV does not leave a residual disinfectant in the water. So it's just light, it doesn't stick around. So once the water leaves the treatment chamber, there's no more disinfection occurring. So a safe storage container prevents people from sticking things in it, hands or scoopers, and there are multiple ways in Mexico, maybe some of you are familiar with them, for how people extract water from these containers that don't involve sticking things in them. So this device was developed over about a 10-year period by a lot of different UC Berkeley students. And one of them eventually, Fermin Regadas, who was a PhD student in the Energy and Resources group, eventually formed a nonprofit organization, shown up here at the top, Fundación Cántaro Azul. And with the help of Cántaro Azul, we carried out a field study in Baja California Sur, which some of you may be familiar with. Here's La Paz here in the middle. And there are all these very rural households located to the north and the south of La Paz, where people don't have access to safe drinking water. So they're collecting their water from local ponds and local springs, but they're not protected. And so they're subject to contamination due to human activities, and also there's a lot of animal grazing. And so the idea was to see if this was an effective way for these households to produce safe drinking water. So you can see from these pictures here that these, especially in this one up on the left, these are very remote households with very, very, very modest households, and they don't have a lot of modern amenities, but within the 10 years prior to us carrying out this project, all of these households gained access to a small photovoltaic array that provides them with a few hours of electricity a day. And so they mostly use it for lighting the kitchen area and maybe an additional living space in the evenings. And if you remember, the Mesita Azul treats 5 liters of water per minute. And so for a family to treat all of the water that they need in a day, they don't need to run it for very long. So they may only need to run it for 15 minutes or 30 minutes a day. So what we found is that households love this technology. So the pictures you see here are actually of a previous version, slightly more artisanal than the final product that I showed you, which was cast out of plastic molded frame. These are actually made out of wood. But people loved them. They thought they were beautiful. They liked the fact that they looked kind of high tech. And in most of the households that participated in our study, when we went back and visited them in numerous rounds of the study, we found that they were prominently displayed in their households and very well taken care of. So people were proud of them and took very good care of them. And so what we found from our study, I'll spare you from all the details, although I've provided references for several of the papers that have come out down here at the bottom. But we found that it was effective for improving the water quality. The only measure of water quality that we're able to implement out in these remote field conditions were measuring E. coli using the IDEX Quantitre method. But it was very effective at improving the water quality, or reducing the presence of E. coli. But we did find some evidence of recontamination. So when we compared water right after treatment with water that had been stored, we found that there was slightly more E. coli in the water that had been stored in some households. And so it's really important to train households to use safe storage practices. And many of these households were not using safe storage before they were introduced to the Mesita Azul. So that's actually an additional benefit of them adopting this technology. We collaborated with Jack Colford's group in epidemiology to actually test whether there was an improvement in childhood health, which we measured as a decrease in childhood diarrhea. And we actually did not find a statistically significant decrease in childhood diarrhea. And we suspect that that's because the prevalence of diarrhea is actually quite low in these communities. The prevalence we measured in our study was between 1.2 and 3.5%, measured over a 7-day period. So households were asked how many household members had had diarrhea in the previous 7 days. So that's great news. Like, it's great that childhood diarrhea is not highly prevalent in these communities, but as a result of that, we weren't able we weren't really able to improve upon that. So we still think that this Mesita Azul technology is very effective at reducing childhood diarrhea due to many pathogens, with the possible exception of adenovirus and other double-stranded DNA viruses, but we weren't actually able to show that in this study, which was also a pretty small study. And that's it. That is everything I wanted to share with you in this lecture. There is a handout. Oh, I think I have a copy of it here. Okay, so I'm also placing a handout that's a summary of disinfection with the 3 sections that we've talked about. So a summary of the mechanism, some important design considerations, and then applications. And so you can go ahead and download that if you want from the B Courses site, and then we will go over that next week in lecture as well. Okay, thank you. See you next week.